Hello and good morning all. Again, I'm your instructor, Dr. Monica Gullianas, and I will be, today we will discuss the next module. We will talk about DNA recombination. So last couple of weeks we were talking about DNA damage and repair, and now we are moving on to the next module. So in this module we will cover DNA recombination, of course, a number of different types of recombination, homologous recombination, site-specific recombination in both prokaryotes and eukaryotes. We will also talk about transposition because that's another sort of another part of recombination. We are focusing on chapter 11 of Watson's 7th edition, and I have given here page numbers that makes most of this lecture and additional materials, of course, and part of chapter 12 as well. And the book has a lot more detailed eukaryotic recombination, but we are not going to go into that much detail. Okay. So recombination, we all have heard of recombination. We all know recombination. Recombination between two homologous chromosomes is a normal normal mechanism that occurs in living cells, and recombination is quite important. You all have heard of crossing over during DNA replication during meiosis, and this recombination, this crossing over events, these bring genetic diversity in a gene pool and a population. So what is recombination? Recombination is the rearrangement of DNA or physical exchange of DNA sequence between the chromosomes. So between the chromosome of the same organism, these chromosomes can rearrange the DNA by crossing over or moving the parts of DNA from one chromosome to another chromosome, homologous chromosomes, not any other chromosome. Remember, we are diploid organisms, so we have one chromosome from father, one chromosome from mother, so a paternal chromosome and a maternal chromosome. These chromosomes, these are in sequences, they are very, very similar. They are not exactly same because we have different alleles for different genes, and these alleles means there is a slight variation between the DNA sequence, and that makes it whether it's this allele is dominant or recessive. But this information can be exchanged between these maternal and paternal chromosomes during meiosis at crossing over, and that's what recombination is. So here in this example, we have these two genes. So remember, these are two genes, one on maternal chromosome, one on paternal chromosome. These are two alleles of the same gene. So in this example, in this female fly here, both of these alleles are dominant alleles in the maternal chromosome and not in the father's chromosome and paternal chromosome. These are both recessive alleles, so it's a heterozygous, it's a double heterozygote here. And when this fly mates with a male which is a homozygous recessive for both genes, so both of these genes are recessive in the male. When these flies mate, so what will happen that this female will make eggs that are either has this allele or this allele, right? So it's either this PRVG or this PRVG here. The male will make only one kind of sperms that all will have the same, because it's homozygous for both of these alleles. So here the sperm will have only one kind of these sperms. However, since the female, so if there is a recombination happen between males, it will not affect anything because these are both recessive. But here, because the female has a recessive allele and a dominant allele, if the recombination happens here, this can exchange the portion between the female and the maternal and paternal chromosomes, and this is what shows here. So this is the parental. This will be if there is no recombination happen, there will be two different kinds of eggs will have these, either of this chromosome with these alleles or this chromosome with these alleles, and sperm will have the the same allele of the sperms. But if recombination happens, then these portions will be exchanged between the maternal and paternal homologous chromosome, and these females now have more diversity. So these populations, these children will have they will be variable from their parents. All DNA in organisms in us is recombinant DNA, because we all of are during meiosis, the cells, the DNA, the chromosomes undergo recombination, and so makes this recombinant DNA. Okay. Now today we're going to talk about different type of recombination: homologous recombination, site-specific, and transposition. In this lecture, I will focus on homologous recombination, and then we will talk about site-specific and transposition later. Homologous recombination is also called a generalized recombination. This happens during meiosis, during meiosis, which is more of a generalized, more of a simple recombination that we talk about. It can also happen between molecules of homologous sequence if replication stalls, as a repair mechanism when there is double-stranded break, and that's what we talked about when we were talking about double-stranded DNA break. There are key steps of homologous recombination, and we already talked a few of these when we were talking about double-stranded break repair, but now I'm going to focus a little more on the mechanism. So the key step of homologous recombination are: first is alignment of homologous DNA molecules. First, the DNA has to find its homologous sequence. So homologous sequence can be identical or nearly identical. And as I just said in the previous slide, that the homologous chromosomes are not exactly same. They are similar because we have different alleles coming from parental and coming from paternal and maternal chromosomes, and these alleles vary a little bit. That's how they make a dominant or recessive allele. So these homologous chromosomes are similar but not exactly the same. Okay, so these the DNA molecule first finds these homologous sequences. That means it finds its correct chromosome and the region of the chromosome where the similar sequences lies. After this will be then will be resection. So that means first will be introduction of breaks. So DNA will have a double-stranded break, a single-stranded break. A nick can also start a homologous recombination, but it's usually a double-stranded break that's more keen or more prone to for making recombinations. And then it will be resection. Of course, there will be the DNA molecules. Molecule will be part of it will be removed to cause overhangs. These overhangs, these 3' overhangs, then leads to strand invasion. That means that single-stranded region of DNA that's generated in the previous step that will pair with the homologous complementary strand in the homologous DNA molecule. Once that strand invades the homologous chromosomes, then it will form the holiday junction. The holiday junction is where two DNA molecules become connected, where it makes that X shape. That's the holiday junction. And once the holiday junction is formed, when the parts have been exchanged and gaps have been filled by DNA pole and ligated, these holiday junctions, before that, this holiday junction need to be resolved so that the DNA becomes two different molecules again, and the genetic exchange is complete now. This is called resolution. Okay. Now here, this is the alignment of homologous chromosomes. So these are two homologous chromosomes. These homologous chromosomes this is the same portion of the DNA. So here you can see that this chromosome has dominant alleles, and this chromosome has recessive alleles, but they're very similar in sequence. So they found this sequence, this similar sequence. And then once they find this sequence, then there's introduction of breaks in DNA. These breaks have been formed by specific enzymes. Once this break is formed, then there will be this resection. That means the removal of the portion of the DNA. This removal of the portion of DNA causes these overhangs or the single-stranded DNA. The single-stranded DNA then invades the homologous chromosome. So this is the homologous chromosome that is going to invade. So this chromosome has a break. It finds this sequence that's similar, then it invades this sequence. This is the Holiday junction where they cross over each other that makes this X shape. And once this is formed, then the DNA pole will fix this portion. That's by using this as a template, this DNA molecule, and then during resolution of the Holiday junction, this Holiday junction will move apart. So here you can see that it's close to this B gene here, and as the Holiday junction is moving apart, it's closer to C, and eventually it will find the right place, right sequence. That's where the Holiday junction will resolve, will break, and that's called the resolution of Holiday junction, and it will result into again two set of chromosomes with where the strands have been exchanged. You can see here that these make heteroduplex here because these are not exactly similar sequence, but they're very, very similar. So there is a little bit of mismatch, but this can tolerate this mismatch. So, yeah. Okay. Now, what's the mechanism of this homologous recombination? As we will talk about E. coli because it's better understood in prokaryotes in E. coli. In E. coli, the homologous recombination is occurred through Rec-BCD pathway. So Rec-BCD pathway. This Rec-BCD it's a complex of three protein subunits: Rec-B protein, Rec-C protein, and a Rec-D protein. This complex has helicase as well as nuclease activity. So Rec-B and Rec-D are helicases, and Rec-C is nuclease. So here you can see this is the double-stranded DNA. It's a 3 to 5 prime strand, 5 to 3 prime strand. This is a chi pointer crossover spot. That's where it needs to come, this complex. This is Rec-D and Rec-B, and this is Rec-C, the nuclease. They bind to this DNA where the break has happened. And then Rec-B and Rec-D and Rec-B will open the DNA because these are helicases. And let's see next. So in presynapsis, Rec-BCD complex will bind to this double-stranded break. It will dissociate the DNA strand because these are helicases. Rec-D is in 5 to 3 prime direction, Rec-B in 3 to 5 prime direction. And the activity of these Rec-D and Rec-B is different, although they're both helicases. This helicase is faster. The Rec-D is a faster helicase, so it's move along faster. And Rec-B is slower. It moves slowly on this 3 to 5 strand. So, however, the activity of Rec-C, the nuclease, is also a bit different for both these strands. It makes fewer cuts on the 5 prime end, but more cuts on the 3 prime end. So as you can see from these arrows, there are 2 cuts here, but 4 cuts here. Since it moves slowly, it makes more. Moves faster, so it probably doesn't need that many cuts. Okay. This Rec-BCD complex, it keeps opening the DNA molecule until it reaches to chi, which is a crossover hotspot instigator. Once it reaches to chi, this activity of these Rec-B and Rec-D changes. What happens is Rec-B will still stay equally active, but Rec-D will have half as activity as it previously was before getting to chi. And now you can see in this diagram here, so Rec-BCD bind to this double-stranded break. They both are opening the sequences. Rec-D is opening it faster, Rec-C is opening it slower. So it's looping around here because it cannot go as fast as the Rec-D. So it's the DNA molecule is kind of looped around here. Once it reaches to chi, the activity reverses, so it becomes a little slower, or it's paused here at the chi, and then Rec-C is the endonuclease will be paused as well. So at this point, at chi, since this portion is, there's some of the 5' end is broken, but 3' end is not as much cut as 5' because this was slower helicase here. So this portion, this loop will open up and it will give that single-stranded DNA molecule. This Rec C recognizes the chi site. Here, this is the chi site. Rec B, this remains active. It pulls the single-stranded DNA loop at this chi square. Here, this is pulling it in because there was lower activity here. Rec D now, as I said, it has reduced activity now, so it will, everything will stay here. It will make this 3' overhangs of single-stranded DNA. So this is the 3' overhang of single-stranded DNA. Once the Rec BCD dissociate from this portion, this will open up and this will be the single-stranded overhang on this end. Now, because it's a single-stranded DNA molecule now, as we all know, that single-stranded DNA needs to be bound by proteins. So the single-stranded binding proteins can come and bind to this single-stranded DNA now. However, during recombination, there is a specific single-stranded DNA binding protein called RAC-A. That RAC-A binds to this sequence now. This RAC-A is a very, very important, it's a central protein in recombination, because it's not only a single-stranded binding protein, but it also is a strand exchange protein. So this RAC-A will promote alignment with the homologous region of the double-stranded DNA molecule, and it will help with the exchange of the strands. Okay, so here again, once a double-stranded break is formed between one of the chromosomes, this chromosomes will go through 5' to 3' resection. That means making these 3' overhangs. These 3' overhangs will then invade into the new chromosome, into the homologous chromosome, and the branch migration and new DNA synthesis will happen here by DNA pole. Holiday junction has been formed here. This holiday junction will be resolved once the sequence is complete. And here you can see if there is no crossover, then it will be just this sequence right here, whatever this was. So, and if there's crossing over happen, then this will cross over. It will exchange the strands completely. Okay. RAC-A, this is that single-stranded binding protein that binds to 3' overhangs. This protein is processive. So it binds in a processive manner, and as you might remember, we talked about being a processive protein or a processive enzyme when once it's one molecule is bound, the multiple molecules will bind. It makes this active filament formation, and it coats the 3' end of single-stranded DNA. So as I said, RAC-A is a central protein in homologous recombination because it's not only coat the single-stranded DNA molecule, but it also acts as a strand exchange protein because it's a member of a family of enzymes that are strand exchange proteins. It catalyzes the pairing of homologous DNA molecules, so that the DNA molecules that it's going to exchange the sequences with. So here, this is the homologous chromosome. This is the one that it's going to exchange its part with. It brings these together and binds tightly to this DNA molecule. There's no base pairing between these strands. It's just this protein is keeping them tightly close to each other. Once these bind together, this other molecules moves away because these two are very close together. It binds cooperatively to DNA, and again, as I said, cooperative binding, we have already studied this. Cooperative binding means once one molecule of protein binds, multiple of those will be bind immediately, just like we talked about when single-stranded DNA binding protein, because RAC-A is also a single-stranded DNA binding protein along with another function. This pairing involves both the search of sequence, because it will only bind at the sequence that's homologous to the sequence on the single-stranded DNA, now the 3' single-stranded overhang. Once it's matched, so it's the protein scans the homologous chromosome for similarity, and once it finds a similar sequence, it will keep those together. the strand exchange will yield in tighter binding by RAC-A. This drives this exchange. The strand exchange between two helices of DNA will require the Holiday junction, because that's the site of strand exchange. That means the Holiday junction that's where they cross over. So here again, this is a double-stranded break. This double-stranded break will lead to RAC-BCD, will generate these 3' overhangs. So RAC-BCD will come and bind here, and it will remove the sequences. The sequence will remove the sequences in a way that the 3' overhang, the 3' will have lower activity of helicase, so it's not as much open and cut as the 5' end, so it will have that 3' overhang. And this 3' overhang, so right here, this 5 to 3' overhang will then invade the strand. So here in this example, this is being shown with this 3' overhang. This is invading the strand. RAC-A binds to the 3' overhangs and mediates this strand invasion. This RAC-A, which acts as both the single-stranded binding protein as well as strand exchange protein. And DNA will be synthesized in these regions. So this is the homologous region. This is the template. So this template will be used to make this sequence. So this sequence might be a little different from its original sequence now, depending on what allele was there on this chromosome. And here this sequence will act as a template, and will be DNA synthesis in this direction. So both now DNA sequences have copied the sequence information from this homologous chromosome, and there will be ligation to close the nicks. Once this is the DNA is complete, there will be branch migration. This another proteins ROV-A and B that helps with this migration of the Holiday junction or resolution of Holiday junction, and ROV-C is the nucleus that will make the cut to resolve these Holiday junctions eventually. This is post-synapsis now. So your pre-synapsis is before Holiday junction formation, then there's Holiday junction formation and exchange, and then post-synapsis is once this information has been copied. So in post-synapsis, RAC-A, which is uses ATP hydrolysis. This ATP hydrolysis removes the RAC-A from single-stranded DNA. Migration is catalyzed by ROV-A. This is the ROV-A protein. ROV-A then also brings the ROV-B together. ROV-A and B, these together exchange the branch. This ROV-A will bind to Holiday junction. This is a Holiday junction specific binding protein. And ROV-A recruits ROV-B, which is a helicase, and which will help open this Holiday junction. And we will talk about ROV-C in the next slide. So the ROV-C really leads to the resolution. The strands will get nicked at Holiday junction. This nick is caused by ROV-C because it's a nuclease. Then this ROV-C ROV-C binds to ROV-A and B complex, along with this that makes this complex with A and B, and these it moves the Holiday junction. It looks for the sequence that has either A or T, two T's, and then either C or G. So that's the sequence, consensus sequence for ROV-C, and that's why this branch Holiday junction need to migrate until ROV-C finds this specific sequence, and then it will make a cut at that specific sequence, and these strands will be apart. So here this is RAC-A, which is strand exchange protein. This helps in exchange of the strand. Second strand exchange, formation of Holiday junction here. DNA polymerase will fill the gap. DNA ligate will fill the nicks. ROV-A and B will come and bind to these position where the Holiday junction is on both ends, and they will move these branches until they will start moving these branches, and then ROV-C comes and cleaves. ROV-C comes and bind to ROV-A and B, and it looks for this particular sequence here. When it finds that particular sequence, it makes the cut. once it makes the cut, these branches will get apart, will get separated, and again the ligase will fill the neck, and now these have been exchanged. Okay, this let me play this video, which is pretty nice, if I can. Yes. So it shows the activity of
one of the major mechanisms to repair double-strand breaks in prokaryotes is homologous recombination. Double-strand breaks in DNA are caused by ionizing radiation and DNA damage, such as lesions and unpaired nicks in the DNA strand. Rec-BCD is an enzyme that contains both DNA helicase and nuclease activities to unwind and degrade the double-stranded DNA to generate single-stranded DNA. The complex uses the energy from ATP hydrolysis to process the activities, and it is controlled by specific short DNA sequences called chi-sites. It is composed of 3 subunits: Rec-B, Rec-C, and Rec-D. Rec-B and Rec-D are the DNA helicases that use ATP hydrolysis to unwind the DNA strands. Rec-B unwind with the DNA in 3' to 5' direction at a slower pace and create a single-stranded DNA loop. Rec-D unwind the DNA faster in 5' to 3' direction. Rec-C recognizes chi-sites and bind tightly to prevent the complex from digesting the entire 3' tails. It helps increase the digestion of 5' tails, and Rec-B reel the single-stranded DNA loop back in. Once Rec-BCD reaches the chi-sites, the nuclease activities increase. Rec-A proteins are strand exchange proteins that play a major role in homologous recombination. They coat the single-stranded DNA by interacting with the Rec-B subunit. Rec-B load Rec-A onto 3' tails. Once they are loaded, Rec-BCD disassemble. Rec-A facilitates strand invasion. Short base pairing between the 2 homologous DNA are formed and heteroduplex DNA is generated. After strand invasion, DNA polymerases fill in the gap by elongating the 3' end of the invading strand. It synthesizes the complementary base pairs to the template strand. There does not need to be a primer to initiate polymerase activity. The invading 3' strand is sufficient to prime DNA for synthesis. The 2 homologous DNA are connected by DNA strands that are crossed to construct a structure known as the holiday junction. The branch migration is stimulated by RUV-A and RUV-B proteins, also known as RUV-AB, to move the junction along the DNA. RUV-A is a site-specific binding tetrameric protein of the holiday junction. It recognizes the junction and binds to it. Then it recruits RUV-B. RUV-B is a hexameric ATPase that supply energy to the base pairs that migrate the DNA branch rapidly in a single direction. The holiday junction is resolved with a dimeric protein called RUV-C. It recognizes and binds to the junction. It cleaves the 2 homologous DNA strands that contain same polarity. RUV-C can cleave both junctions at the same time. The strands are ligated to create patches. RUV-C can also cleave one junction at one site and the other junction at another site. Splices are formed.
Okay, so that was prokaryotic recombination, homologous recombination. Now we will talk a little bit about eukaryotic homologous recombination. Eukaryotic homologous recombination is very similar to the prokaryotic mechanism that we just talked about, but there is no Rec-BCD pathway. It works in a little different way than what we talked about in prokaryotes. So again, the initiation requires a double-stranded break, which is followed by a 5' to 3' exonuclease activity to create the single-stranded DNA. So these SPO11 and MRX are very important to make these double-stranded breaks. SPO11 is meiosis specific, so during meiosis, this SPO11 will make the double-stranded break. MRX can make double-stranded breaks with both meiosis and mitosis, usually mitosis. So these will bind to, so they will make the double-stranded break and stay bound there. So this double-stranded break formation by SPO11 here, and SPO11 is bound. So once this break is formed, and the other portion of the mechanism, other part of the mechanism are activated, this SPO1 will disassemble, will dissociate, and then MRX, this 5' to 3' resaction, will be carried out by the MRX. So here these are MRX proteins. These will lead to this 5' to 3' resaction. Then once this 3' overhangs are formed, MRX will be removed, and DMC1 and RED5 will come and bind. So RED51 is similar to REC-A of eukaryotes, and it's a strand exchange protein. So here, this double-stranded DNA, once first the double-stranded break are made, these double-stranded make then leads to the resaction, 5' to 3' resaction, which makes these single-stranded DNA templates or single-stranded overhangs. This results, then it will go and it will find the homologous sequence on the chromosome, on the homologous chromosome, and this homologous chromosomes will then be invaded by these 3' overhangs. This invasion will form the holiday junction, just like we talked about in the eukaryotic chromosomes. These double-stranded break are purposely caused during meiosis by SPO11, because during meiosis this is a common thing for DNA to recombine in order to bring genetic diversity. SPO11 is an enzyme which is similar to topoisomerase. You can also think of HO when we were talking about the yeast, where HO is an endonuclease which is specific for that particular purpose, for mating switches. This digestion of 5' ends is mediated by MRX, so these proteins will do that, and then REC-A function is provided with this RED51 in mitosis and meiosis, and DMC1 only during meiosis. So DMC1 will only function in meiosis, but RED51 will function in both. MRX protein, I'm sorry, I said that was functional in mitosis. That's not the case. MRX is more of a protein that's more equivalent to the function that REC-BCD pathway does in prokaryotes. Okay, so now if we look at these different steps in recombination, or different proteins that are involved in recombination steps, so pairing of homologous DNAs and strand invasion is carried by REC-A protein in E. coli and RED51 in eukaryotes. DCM is only in meiosis. Introduction of double-stranded breaks is, we don't know what causes these double-stranded breaks in E. coli, but in eukaryotes it's a SPO11 which causes these double-stranded breaks in meiosis. HO, as I mentioned, it's also was very specific, that causes double-stranded breaks in mating type switching in yeast. Processing of DNA breaks to generate single strand for invasion is caused by REC-BCD, helicase of nuclease in E. coli, and MRX protein, which are also called RED50, 58, or 60 nucleases in eukaryotes. Assembly of strand exchange proteins as REC-BCD in E. coli, and RED52 and RED59 in eukaryotes. Holiday junction recognition and branch migration is caused by RUF-AB complex in prokaryotes. It's not well characterized in eukaryotes, we don't know very well. And resolution of holiday junction is carried by RUF-C, which is the ortholog to RUF-C in eukaryotes are RED51 CXR CC3 complex. Okay. So consequences of homologous recombination. The main consequence of homologous recombination is to generate genetic diversity. This genetic diversity is important for a population because more diverse the population, the individuals will be better adapted. They will have better chances to survive. This recombination initiates between identical sequences at chi-square or chi-sites. These, but they can involve strands that are similar but not identical, and that's because the homologous chromosomes are very, very similar, but they're not identical because they carry different alleles of the same gene. This can be used for as a tool for genome editing that we have talked a few times already during tools and techniques and during double-stranded break repair as well. So genome editing uses this pathway in order to cause very specific changes in the genome. These changes can be very subtle or can be more bigger changes, but these are very specific. These are not random changes. And this genome editing tools, they harness this natural repair mechanism to modify DNA. Again, we make this double-stranded breaks. Well, instead of radiations or chemicals that cause random mutation, we make this very sequence-specific double-stranded break. Say, for example, in CRISPR, we use guide RNA and Cas9 to make these double-stranded breaks, because guide RNA will take it to where we want to make the break, and Cas9 is an endonuclease that will cut the DNA. Then it can use either two pathways, homology-directed repair that we just talked about. What that would do is it will use this specific template in order to fix that double-stranded break. It can correct mutation. If there is a mutation in the sequence, we can use a sequence that is correct and correct that mutation. We can introduce mutation to see what that gene does, or we can insert a gene if, say, for example, if it is lacking the correct allele, or if we want to have a gene, an allele that's or a gene that's is beneficial for that organism. So we can introduce those genes. Say, for example, in plants, we insert genes that make them resistant to insects, resistant to salinity or high temperature. So these are all the beneficial mutations that we can make. Another way of genome editing is non-homologous end joining, which just causes random indels and knock out a gene. Okay. So this is another video that if you want to watch later, this kind of just summarizes all that what we talked about in terms of homologous recombination. Okay, and with that, this is all about homologous recombination that we're going to talk about. Then next we will talk about site-specific recombination and transposition. Okay, so there will be a quiz today, and I will talk to you again on Wednesday. Okay, take care.